James did his undergraduate at UC Berkeley and then came here to Michigan to do his PhD in the Department of Earth and Environmental Sciences. He actually completed his PhD earlier this year and is currently working as a postdoc researcher at the University of Michigan Museum of Paleontology. And later this month, he'll be starting a new position as a postdoc fellow at the University of Oslo Natural History Museum, where he's going to be studying rates of evolution. James is interested in analytical paleontology, invertebrate zoology, and evolutionary biology broadly. And today he's going to be talking about dispersal from the West Tethys to the modern marine diversity hotspot. Take it away, James.
Okay, thank you for that intro. And thanks for that nice talk, Jake. Can everyone hear me okay? Like a single thumbs up would be all I need. Thank you. Hey all. So I finished my PhD this year with my co-author on this talk, Tom Baumiller, and I'll be presenting some of our findings on the biogeographic history of some marine invertebrates. And I'll be synthesizing that, or trying to synthesize that with some evidence from other groups to try and make sense of the center of diversity in the oceans today. So a good chunk of ecology and evolutionary biology worldwide seems to be devoted to understanding and explaining why diversity is unevenly distributed around the globe, and especially to explaining concentrations of great diversity, or also known as hotspots of diversity. There might not be much to explain here if every group showed kind of an idiosyncratic and a different pattern. But what's interesting is that different clades often show strikingly coincident patterns in global diversity, the latitudinal diversity gradient probably being the most famous of these. In the marine realm, the Indo-West Pacific biodiversity hotspot is the most conspicuous center of diversity of this kind. That's shown here for groups as disparate as corals, mangroves, seagrasses, and bivalves. Explanations for centers of diversity like this frequently invoke aspects of diversification, things like elevated speciation within the hotspot or elevated extinction outside of it. When dispersal or migrations enter into these kinds of global theories of biodiversity, they typically act to move lineages from regions of high to low diversity, sort of acting like pressure accelerating particles from regions of high to low pressure. By and large, though, diversity is typically conceptually linked with diversification, high diversification. A complication in this sort of inferential or explanatory scheme has arisen in the last half a decade or so, as phylogenetic studies of diversification have frequently found that some major groups, including brittle stars, fish, some birds, and plants, some plants, diversify at their greatest rates outside of their regions of greatest diversity. In Jake's talk, you heard some interesting and really worthy issues in interpreting these kinds of studies, but at least they suggest that other sorts of explanations may be worth considering when we're trying to explain these centers of diversity. I'll be considering a case in which the center of richness appears to owe its existence not to elevated diversification, but to invasions from outside of the hotspot. I'll try to show that this group that I'm working on is not exceptional in this regard, and I'll consider what that would mean for theories of global biodiversity very briefly, and I hope I don't go over time. The first thing to know about the marine richness hotspot is that it is geologically young. That's shown here with counts of fossil genus richness in large benthic foraminifera, which show that from the Eocene to the modern, the center of greatest richness has shifted from the West Tethys, so the western end of this great Tethys seaway. Let me see if I can get my laser pointer on. The western end of this great Tethys seaway, through the Arabian Peninsula, and finally to reach the Indo-West Pacific sometime between the Miocene and today. This has been supposed to attract tectonic activity. So the collision of Africa with Eurasia is thought to have destroyed a lot of shelf habitat in what is now the Mediterranean. And at the same time, roughly, the collision of Australia with Southeast Asia is thought to have generated a lot of new shallow habitat in that area. So this pattern of a shifting or hopping hotspot has become increasingly clear over the last decade or so of research, with fossil and phylogenetic support from other groups generally tending to confirm this pattern. But no clear consensus has emerged on the processes by which this pattern takes place. And by that, I mean that different combinations of speciations and extinctions and dispersals, or rates of these processes, remain largely unconstrained or plausible as factors that have moved this center of biodiversity. In trying to work this out, raw counts of richness are limited in what they can tell us about these processes. So, for example, plots like this are equally consistent with mass migration of lineages from west to east, and equally they're as consistent with that as they are with, for example, intense proliferation of lineages in the new hotspot. I've been trying to work this system out in a taxon that I've spent a lot of time thinking about, the Comatulid crinoids. These are a group of benthic, suspension-feeding marine echinoderms. They look like this, very handsome group of conspicuous tropical reef denizens. They show up in the early Jurassic in the fossil record, and today they comprise most of the diversity of this ancient group known as the crinoids. Might be familiar to some of you. Comatulid crinoids occur at all depths in the sea and worldwide. They are quite mobile, so they can move around and swim in the water column for minutes at a time. They have, like a lot of marine invertebrates, they have larvae that persist in the water column. They don't persist for very long in the water column, but the comatulid populations seem to show substantial genetic connectivity across great distances, and so we tend to think that they have a good ability to disperse around the globe. In any case, like other groups, they show this Indo-West Pacific hotspot, and not just in terms of taxonomic richness. That's shown here on the left with species counts in each 10-degree cell of the globe, with data downloaded from Ocean Biogeographic Information System. But this region, the Indo-West Pacific, also seems to be the greatest concentration of evolutionary history for this group. So in this, the first detailed study of the historical biogeography of the Comatulid crinoids, I and my co-author Tom have used ancestral range reconstruction, a new fossil data set, and some novel preservational analyses, plus some new fossil phylogenies to try to work out what processes may have underlain the origin of this center of richness, center of diversity. I'll jump first into the evidence from living Comatulid crinoids. Here I'm showing you a molecular phylogeny of this clade. Sorry for this chaotic pointer. A molecular phylogeny of 139 species of Comatulid crinoids. This phylogeny was generated with an alignment of 4 molecular loci, something like 6,000 base pairs long, inferred with likelihood, maximum likelihood as implemented in MaxML, and timescaled using penalized likelihood using 3 fossil calibrations. I scored every living species as being present in either the Indo-Pacific, the Atlantic or Mediterranean, those are in black and white respectively, or as being widespread in gray. And I inferred biogeographic history on this phylogeny using the DEC model of geographic range evolution. This model is similar to the perhaps more familiar MK model of phenotypic character evolution, but it differs in how it treats polymorphisms in a way that makes sense given how we think geographic range evolves. And in this inference scheme, I allowed rates of dispersal to vary. So I allowed the rate of dispersal into the Indo-Pacific to be different from the rate of dispersal out of it. So let's see what that looks like. The best inferred model is shown here. At each internal node, I've shown the relative likelihood of each possible ancestral state, ancestral range at each node. And what you can see is that even though most of the tips in the phylogeny are present in the Indo-Pacific, reflecting their diversity hotspot there, most of the basal nodes in the tree, including the root, are inferred to have been in the Atlantic or Mediterranean. This has something to do with the fact that the inference scheme inferred asymmetrical dispersal rates. That's visualized here. On the x-axis, I'm showing the ratio of dispersal rates out of versus into the Indo-Pacific, with high dispersal out of the Indo-Pacific on the right here, and high dispersal into the Indo-Pacific on the left here. In the top panel, I'm showing the likelihood of reconstructions using each set of parameters, and on the bottom, I'm showing the corresponding reconstruction of the ancestral range at the root, given that ratio. And what you can see is that the maximum likelihood estimate of this ratio estimates a relatively high rate of dispersal into the Indo-Pacific, something like 23 times the rate of dispersal out of it. Correspondingly, the inference scheme infers an Atlantic Mediterranean origin. This asymmetrical scheme adds an extra parameter and so kind of an epistemological burden, but this burden of the extra parameter is more than justified by the boost in likelihood gained by inferring asymmetrical rates of dispersal. To translate, the best explanation for the data from living comatulids is that this clade originated in what is now the Atlantic or Mediterranean and dispersed at a relatively high rate many times independently into what is now the Indo-Pacific. So that's all well and good, but this sort of inference scheme is inferring a lot of history from a single time slice, and it might be interesting to find out the degree to which this can be independently verified with fossil evidence. So let's take a look at what the fossils tell us. Tom and I built a new database of fossil comatulid occurrences, something like 400 occurrences, and that's shown in this figure, showing paleomaps from the early Jurassic to the Pleistocene. In black crosses, I've shown the locations of fossil localities that bear fossil comatulids. And in circles, in dark circles, I've shown the locations of localities that bear other kinds of echinoderm fossils. These are used as a preservational control, which I'll explain in just a sec. These crosses show a gradual geographic expansion of comatulids from an origin in the early Jurassic of Western Europe. A slow expansion out from there with a first appearance in the Americas in the early Cretaceous. And the clade does not appear to appear in the Indo-West Pacific until the Oligocene, and from then on they're widespread. This, on the surface, seems to match what was inferred from the molecular phylogeny. But obviously the fossil record is imperfect and unevenly sampled, and we might not feel comfortable taking it at face value, so reading it as a literal record of biogeographic history. For example, we don't find any fossil comatulids from the Pleistocene of Antarctica, and we know they're there today. So if we can't read it literally, can we make a more limited inference from this data set? Can we infer that this clade originated in the Western Tethys and only reached the Indo-Pacific much later? In order to get at this question, I used the idea of a preservational control. This is a group which has some similarities preservationally or ecologically to our focal group, the comatulids, and whose presence indicates suitable depositional conditions for the preservation of our group. In this study, I used fossil echinoderms as this sort of a preservational control in this sense, downloaded from the paleobiology database. And I tested the degree to which we can read the comatulid record as a literal or as containing real biogeographic information using two new quantitative approaches. The first uses a sort of a model selection approach, and the second uses an approach using geographical extent. So let's look at those now. First, I'm just showing the number of echinoderm and comatulid bearing localities in the Western Tethys and the Indo-Pacific for each of 10 intervals from the early Jurassic to the Pleistocene. You can see that comatulids are initially absent from the Indo-Pacific, and we want to consider whether this absence is a real biogeographic signal or if it just reflects insufficient sampling to have found them there yet. You can also see that our preservational controls, the echinoderms, non-comatulid echinoderms, are, for most of these time intervals, more well represented in the Western Tethys, but they're always there to some degree in the Indo-Pacific. So we always have some window on that region of the world. In our first test, we assumed that our first test relies on the assumption that every echinoderm-bearing locality has some chance of yielding fossil comatulids. In this approach, I compared two models, which I called equal chances and unequal chances. Under the equal chances model, every taphonomically appropriate locality has an equal chance of yielding fossil comatulids. And in unequal chances, Western Tethyan and Indo-Pacific localities have different chances. We can compare the predictions of these two models with the empirical number of comatulid localities in each region for each interval and use model selection to consider them both. I'll show the results of this first just for a single interval in Middle Jurassic. In gray here, I'm showing the probability of observing some number n Indo-Pacific comatulid localities as predicted under the equal chances model. And what you can see is that if every echinoderm locality, every taphonomically appropriate locality had an equal chance of yielding our focal group, we should expect to have found 3 or 4 comatulid-bearing localities in the Indo-Pacific by now. And the likelihood of observing 0 is actually quite low under equal chances. And so for this interval, model selection using Aki-Iki weights overwhelmingly favors a model in which Western Tethyan localities are more likely to yield comatulids. We find similar results for a lot of their early history, of this group. From the Middle Jurassic to the Eocene, we observe fewer Eastern Tethyan comatulids than predicted under equal chances. Instead, model selection favors models in which Western Tethyan localities are more likely to yield these fossils. This is true until the Oligocene and the Pleistocene, and again in the Pleistocene. So given some assumptions, we can cautiously reject the premise that the absence of comatulids from the Indo-Pacific during the first part of the record is just the result of poor sampling there. It seems to reflect some real biogeographic information, genuine biology. In our second analysis, a little simpler, I considered whether the gradual increase in the geographic extent of fossil comatulid localities, again, represented a real biogeographic phenomenon or could be explained just as a result of the vagaries of differential sampling through time. To do this, I compared the geographical extent of fossil comatulid localities in black with the extent in terms of convex hull area of echinoderm localities, our preservational controls, subsampled to the same sample size for each interval. The results are shown here. So in black, you can see that the extent of comatulid-bearing localities increases monotonically from the early Jurassic to the late Cretaceous, and from the Eocene onwards is greater than any point previously. Moreover, from the Jurassic to the Paleocene, this extent is lower than would be expected from just randomly subsampling taphonomic control localities, preservational control localities. What this indicates is that, again, rather than just following sampling, this record seems to be telling us reflecting a real geographical expansion of these comatulids through time. It tends to support, in fact, a Western Tethyan origin for the comatulid clade and a subsequent, maybe Cenozoic expansion into what is now the Indo-Pacific. So that's the biogeographic pattern, but without any kind of a phylogenetic information, it's hard to get a process from that. So that record that I just showed you is consistent with a story involving intense dispersal from the West Tethys eastwards, but it's equally consistent with a story involving intense diversification in the new hotspot. So to try to make that leap, I inferred the phylogenetic affinities of several Mesozoic fossil comatulids using a morphological matrix of 30 discrete and 24 continuous characters. I inferred relationships using maximum parsimony as implemented in TNT, and I constrained relationships among living comatulids to be equal to the relationships observed in the molecular phylogeny. So all I was inferring was the placement of fossils on this scaffolding or backbone. what you can see is that all fossil groups came out nested more or less deeply in the crown of Comatulida. Placements are roughly similar for discrete and continuous subsets of the data. And what this tends to indicate is that the Western Tethys apparently included representatives of some quite derived crown lineages, apparently before the group even became established in the Indo-West Pacific in the Cenozoic. This implies a considerable diversification of this clade before they appeared in the Indo-Pacific, and in fact necessitates a number of independent dispersal events eastward, if we take this phylogenetic assignment seriously. In this respect, it basically agrees with the results of ancestral range reconstruction, which again indicated an origin outside of the Indo-West Pacific and a subsequent elevated rate of dispersal into it. Are migrations the only thing responsible for generating this new hotspot? Maybe not. There are some good reasons to think that extinction outside of the hotspot may have played an important role in the history of this clade, especially in the Mediterranean. Sometime in the Miocene, there was a local extinction event known as the Messinian salinity crisis, basically acting as sort of a reset on life in what was once the West Tethys. So that's a local extinction event that probably played an important role in causing this group to be relatively so diverse in the Indo-West Pacific. As for a role for speciation, elevated speciation in generating this hotspot, what phylogenetic information we have doesn't suggest any obvious imbalances or bursts of origination, but at present it's inadequately sampled to speak conclusively on this issue. In summary, phylogenetic and fossil evidence from this clade tends to support a scenario of intense dispersals into the Indo-West Pacific and perhaps intense extinctions outside of it. And in this respect, they show some interesting agreements with data from other taxa. So the invertebrate record shows clearly that there were seemingly some local extinction events in the Caribbean and in the Mediterranean during the Cenozoic, and we know of nothing like that from the Indo-West Pacific. And moreover, many fossil taxa seem to appear outside of the hotspot before they appear in it. And some phylogenetic studies, for example, of corals, is a nice 2018 study that looked at this, have tended to recover intense rates of dispersal into the hotspot. It's been suggested that this intense dispersal into the Indo-West Pacific may have resulted from changes in oceanic circulation that would have caused propagules, larvae, dispersers to tend more to be swept into the new hotspot. This seems possible, but we see no problem with the perhaps more parsimonious explanation that the creation of new shallow shelf habitats in the Indo-West Pacific would have made invasives more likely to become established in this new hotspot. It's been suggested that the Indo-West Pacific may have served as sort of a center of speciation, either through some sort of a glacial-eustatic mechanism with repeated drops and rises in sea level acting as a sort of a speciation engine, or through just the kind of the topography of the region with isolated islands separated by vast stretches of deep sea, that these might have combined to make it a center of speciation. Importantly, though, the biogeographic evidence doesn't seem to support this. The region hosts a lot of old species, but especially it hosts a lot of species with broad geographic ranges that extend across these isolated island systems, seemingly not really caring that that populations within species are separated by vast distances of deep sea. The region also hosts low rates of endemism, low rates of endemics. And as far as I'm aware, no phylogenetic study has conclusively demonstrated a high rate of speciation in the Indo-West Pacific. What this all adds up to is that the center of richness increasingly seems to exist not because of local conditions that promote proliferation of lineages, but more likely because of its local conditions and its proximity, especially to the Western Tethys, making it well situated to take on the biological inheritance of this more ancient center of richness in the Western Tethys. So with that, I think I'll just wrap it up and we can take questions from me and Jake. Thank you.
Thank you. Thank you, James, for that great talk. If anyone has any questions for James or for Jacob, feel free to either put it in the chat and I can read it out loud, or you can just speak up and ask the question yourself.
James, if no one else is going to ask a question, I'll ask you a question. You've mentioned the increase in shallow shelf area in the Indo-Pacific hotspot during the plate collisions. What about depth gradients? Because I know there are very strong depth gradients as well. So I'm curious with the comatulids, are they primarily shallow water species, or do any of them occupy some of these strong depth gradients in the Indo-Pacific region? I'm just wondering how much that as a factor might also be in hosting many species, not just of comatulids, but some of the other groups that you've mentioned.
Yeah, that seems to be a sort of a, I don't know about a blind spot, but an issue that's hard to get at, like what deep sea diversity is doing during this time. Like it's so easy to think about, you know, just the shallow water biome as being representative of what's going on in the ocean. But I mean, for a lot of groups, like diversity seems to peak at quite deep depths of like a kilometer or two below the surface of the ocean. I feel a little lost thinking about how that sort of depth gradient would complicate thinking about this in a paleontological framework, since most of our fossils are going to be from shallow water, from shelf depths. I do feel like the comatulids are a decent group for looking at this, though, because they don't seem to care about depth in the way that some other groups do. I mean, there are clades like, I'm thinking of clades like bryozoans, where they seem to really favor shallow water over deeper depths. But comatulids, it's not infrequent for taxa that occur in the intertidal to also be present, you know, below a kilometer or two in depth. And so I would like to think that what we find for shallow water wouldn't be too, you know, conceptually or empirically different from what we would find if we could look into the deep history of the deep sea. But I don't know. I mean, I hope I'm wrong.
Thanks.
Thank you.
I'll jump in unless somebody else has a question. So I was thinking about fish, and you know, actinopterygians in particular really blow the roof off in terms of their diversity in the tropical Indo-Pacific. It's really high, and it's not even remote, even compared to like the shallow water Caribbean. And a lot of the diversity does seem to be, while there's clearly no signal of fast speciation rates, it does seem to be in situ derived, and it's very shallow water, you know, upper 100 meters, clearly shallow water, coral platform communities. That's what most of the diversity is. And if you look at like, you know, butterfly fishes or angel fishes, or, you know, kind of go down the list of like classic coral reef associated clades, which is the vast majority of the diversity, I would, you know, there are obviously, the diversification has happened, I think, there for a lot of the groups. I was just wondering how you think about the story for fish fits in to some of the other groups where you actually have some, you know, paleontological evidence on what may be going on, if you think things are similar or different or what.
Thanks. Yeah, I don't know. I mean, obviously the record is less good. It seems like most of the people who have been trying to think about the hotspot with regard to paleo have relied pretty heavily just on Monte Bolca and maybe a couple other bits of fossil stuff. But at the other end, they have, you know, vastly superior, fish people have vastly superior resources in terms of phylogenetic information. There's a paper from, I think, early this year, maybe March or so in PNAS, looking at, I think some sort of a GeoSSE modeling maybe with fishes. They found some results that I had some trouble chewing on. They found that, if I'm remembering this right, they found that dispersal rates for some guilds of fishes were higher outs. So like rates of dispersal from within the Indo-Australian archipelago were higher from within it to outside of it than in the opposite direction, if that's lucid. But I reran their analysis and using a coarser binning scheme in which I just considered Indo-Pacific and Atlantic Mediterranean basins, and I found the opposite result. In other words, higher rates of dispersal from the Atlantic into the Indo-Pacific. I guess I'm confused by these results, and I don't really know how to interpret them. It seems like this sort of an asymmetrical dispersal scenario might be hard to really. There might be some erasure of information, or it might be hard to actually recover what's really going on, and that's why I guess I kind of reflexively fall back on the fossil data. But I guess I don't know. I mean, they're kind of like the main group that people look at this stuff with. So I imagine more stuff will come out on that in the coming years. Sort of a half answer, but thanks.
If no one has any other questions, we're at 5. So we'll give one final round of applause to both our speakers. This was a great way to start off our seminar series this year.